Hello everybody, welcome back. This is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and let's continue our discussion about the artificial way of doing the vegetative propagation. So, in this series of artificial vegetative propagation, first thing what we are going to discuss is about the cutting. So, in the case of cutting, what we have is that you can actually use the different plant parts. So, we have plant has three different parts, one is stem, one is root and the leaves, and you can cut these part into multiple small fragments and then you can put them into the soil, and each of these individual fragment will give rise to the new plant. How it happens? It happens that you have actually the buds on each of these parts, and these buds actually give rise to shoot on the top and as well as the root at the bottom. Let us take an example how it happens in the stem. So, what happened that you have a stem, okay, and it has the joints, and these joints are called as nodes, and on these nodes you have the axillary buds. Okay. So, once you take this stem and put it into the soil, okay, this node part which contains axillary bud is below the soil, okay, and then it starts developing the roots on one side, and this shoot it started coming. Okay. So, this shoot started coming and then you started developing the plantlets along the, and so this is the example of root. Exactly the same thing can happen with the different plant parts, and the classical examples are, in the case of stem you have sugar cane, you have rose, you can have grapes. Okay. Similarly, in the case of root, you have an example of tamarind, and then you have the leaves. In the case of leaves, you have an example bryophyllum. Okay. So, as I think if you remember in our previous lecture also we have discussed how the bryophyllum leaves have the axillary bud on the leaves, and these axillary buds if you put it into the soil they will give rise to the new plant. Now, after this the second mode of artificial vegetative propagation is the layering. Okay. What is mean by layering is that you actually, so you can actually make the physical damage into the stem, okay, and that physical damage actually will become a stress condition for the original plant, and as a result it actually develops the root on the other side. Okay. And you can do actually the layering by two ways. One is called as the mound layering, okay, and the mound layering, the classical examples are the jasmine. Okay. In these cases, in the mound layering, what you do is actually imagine that you have a plant, okay, with leaves and other things, okay, And what you can do is you can take the, so this is the soil on which you have a plant, and in this case what you can do is you can take the branches which are closer to the soil. So, suppose you have taken this branch and this branch goes straight, okay, like this, okay. So, you this since this branch is closer to soil, what you can do is you can take this branch and dump it into the soil, okay. Okay. So, as a result what will happen is some portion of this root or some portion of this branch will below the soil, and this portion will contain the auxiliary buds. Okay. So, they will develop the roots on one side and then they will start developing the shoots on the other side, and after this happens you can cut the plant from here, the original plant, and then this will become the new plant or the plantlets. And this is the way you can actually keep going. So, this is actually a popular method for all the plants which are where you have the branches at the closer end, okay, and where the branches are weak enough so that you can bend them and put them below the soil so that they can develop the roots. But in the cases where you do not have such privileges, for example, in the case of trees, the trees, even if they have the branches at the soil surface, you may not be able to bend them and put them under the soil. So, for those cases you do another way of layering, and that is called as air layering. In the air layering, what you do is so you can imagine that this is your tree and this is the branch. Okay. So, in the and that branch contains, so this is the thick bark which is present on this, and you can imagine that it has leaves and everything. Okay. So, what you can do in the case of air layering is that you just remove the small portion of the bark, okay, along with the tissue. Okay. So, you are going to have this, there will be a gap. Okay. So, you just generate this a gap, okay, and on this portion what you do is you just cover this whole portion with a soil, okay, and then ultimately you cover this whole portion into a polythene bag. Or, so this is the polythene bag and this is the soil and this is the gap or the air or the layer. Okay. So, as a result what will happen is after a couple of weeks this portion will start developing the roots. And this root, then you can see these roots when they come out from the soil, okay, and that is the time when you can actually cut this branch from this side and put them into the pots. Okay. But since these roots system is still not fully developed to support the complete branch as well as the leaves and other things present on this branch, you might have to cover this whole plant with polythene, so that just to maintain slightly aseptic conditions as well as to protect the to give more support to this immature plantlets. The examples of air layering is the rubber plant, the orange, and upper. So, these are the two methods where you are actually using the original plant and generating its multiple copies. But what happens is, in these kind of methods, whether it is cutting or layering, you are not being able to have a flexibility of making the changes into the quality or the quality of the plant. So, in those cases where you are actually trying to mix the qualities of two different individual plant, you go with the another technique that is called as grafting. So, as I said, the grafting is just like generating the chimeric plant, but this is actually a traditional way of making the chimeras, okay, where you may not know anything about the genetics, and it's all chimeras are not being developed at the genetic level. So, in the grafting, what you have is as I said, the grafting actually works with two different species with the plant. The only thing common between the species, only the common between the plant 1 and plant 2, only thing which is common between the plant 1 and plant 2 is their vascular system. So, you cannot use the two plants of different vascular system, and when I say the vascular system means you can take the two plants of a same species with the similar vascular system, means the arrangement of the xylem, phloem and cambium should not vary between the two plants, because in this case you are using one plant to provide some features and the other plant you are using it for the some features. Okay. The plant which actually going to work at the bottom is called as the stalk, or the plant which is going to provide the nutrition in terms of water is called as stalk. The plant which is going to give you the nutrition in terms of sugar is called as scion. Okay. So, scion will be on the top portion, the stalk will be at the bottom portion, and the purpose of taking an stalk or the scion is that scion may have some features which are very advanced. Okay. For example, there are plants which is a plant which where that has a very high level of productivity, okay, or the size of the fruit is very big. So, those kind of features we can take for the scion, but those plants vascular system is very bad. Okay. So, they cannot provide enough nutrition, or they cannot provide the enough stability to the system to hold those many fruits. So, for that you are using the stalk. So, what happen is you take a stalk, means this is your original plant, and on this you add the scion. Okay. So, in this case, since this stalk is good in providing the water as well as the natural resources like minerals and other things, but the fruit which is coming out from the stalk is not of very high quality. Okay. So, what you do is you take another plant as a scion and put it into this stalk, and that actually will give you a good quality fruits. Classical example is different varieties of mangoes. Okay. So, there are different ways in which scion can be put onto the stalk, and that is how we have different varieties of different ways in which we can do the grafting. What are these different ways? Number 1, whip grafting. So, in the case of whip grafting, what you do is you take a scion, okay, and cut it on a particular angle. Similarly, you take, so this is the scion, you cut it obliquely, okay, whereas similarly you take your stalk, okay, and cut it on the complementary mode, and these two you just tie up, okay. So, this is your scion, this is the stalk, okay, and then you tie up this joining point, and yes, you just allow the vascular system of stalk and scion to mix together, and then ultimately you are going to have the new variety of the particular plant. And there are several examples of mangoes, there are several examples of guavas, and there are several other examples where this technique is used. Then you have the wedge grafting. So, what is there in the wedge grafting? So, in the case of wedge grafting, what you do is you make a pointed cut onto the scion, okay, and then you take your stalk and make a notch like this, okay, and then you put this here, okay, and you either can tie up or you can cover this whole thing with wax, and that is the method by which you can do a wedge grafting. Number 3, you can do a crown grafting. In the crown grafting, you do exactly the same way, but you put the multiple wedges on the single stalk, and that's actually is called as crown grafting. Then you have the side grafting. In the case of side grafting, you take the stalk, okay, make a cut here, okay, and then you put your stalk here, tie up, okay, and this is called as side grafting. So far what we have discussed is that you are actually can take a branch of the good quality scion, and then you put it onto another plant of wild type or less good quality stalk, and that actually will give you the better quality plant in terms of economical values, in terms of giving you the good fruits, or in terms of the fruits which are resistant against against the infection and other things. Now, we have another type of grafting, and that is called as bud grafting. So, in the case of bud grafting, instead of taking this whole branch, what you do is you take a part of the bud along with the small piece of cambium, so that it can actually connect it to the vascular system. So, what you do is you take the bud, so this is the apical bud along with a small portion of the cambium. You take your stalk, okay, make a wound, okay, in the middle, okay, and then you place it here. So, you place it your bud. So, this is the bud you have taken out from a particular plant, and then you put it into the center of this particular stalk, and then you tie up this, okay, and then you when you leave it, what will happen is this bed is going to get connected to your stalk and it will give rise to the new plant. Classical example is rose, apple, peach, etc. Whereas in the case of side grafting, the examples are mango, apple. Okay. So, this is all about the very ancient way of doing the artificial vegetative propagation. Apart from this, we also have the artificial vegetative propagation, where the small part of the plant can be used to regenerate the whole plant, and this new technique is known as plant tissue culture or the micropropagation. So, in the case of plant tissue culture, as you can see on the given slide, it involves several steps, okay? And it involves almost 8 different steps, and where you start with a small portion of the plant, which is you are maybe having in the pots. So, first you select the explant, then once you selected the explant could be leaf, explant could be root, explant could be stem. Then you do a surface sterilization of the explant, means you actually remove the bacteria and other pathogenic organisms. Once you are done with that, then you inoculate the explant into a sterile media, and that media you will prepare beforehand, and then that media contains different type of components that actually promotes the growth of the explant to form the callus. Callus is actually nothing but the undifferentiated mass of tissue, and that callus is then being differentiated by changing the composition of media either to root or shoot or the both, and ultimately once you have the root and shoot on the callus, you are actually going to get the small plantlets. These small plantlets then you can acclimatize and transfer it to the plant, transfer it to the pots and transfer it to the outer environment, and that actually is the step called as hardening, and that is how you actually get, you can actually get a plant from any plant part. Okay. So, for this doing this, you have a very specialized set of equipment or the instrument what you need. So, you need a instrument which actually can be used to cut the explant. You need the forceps, so that you can manipulate the explant and you can do the procedures. Then you also need the scalpel to cut the different types of plants. Then you also need the different types of petty plates to process, and then you need the Whatman filter paper also. So, let us see what are the different steps involved in plant tissue cultures. So, the first step is to prepare the media, and then you prepare the media and then do the sterilization. Then you select the appropriate explant and then do the surface sterilization of the explant. Then from the explant you prepare the disc. In this case, in this particular example, we have taken the explant as the leaf. So, that is why we are using the disc preparation, and then you inoculate these discs into the media and allow them to form the callus. Once the callus are formed, then you incubate it for different methods, different media to develop the root and shoot, and once the root and shoots are developed, then you let the plant to be acclimatized into the new environment, and ultimately you put it into the pot and put it these pots outside the in the outside environment to become the for hardening. So, let us see what are the components you need for preparing the media. What you need is the MS media for making the plant tissue culture media. Apart from the MS media, you also need the major salt, you need the iron, you need the minor salts. The composition of major salt is the ammonium nitrate, potassium nitrate, calcium, magnesium, and other kind of thing. You also need the iron to support the growth of the chlorophyll as well as the other green pigments present in the plant. And then you also need the minor salts. These are minor salts could be iodide, bromide, manganese, zinc, and molybdenum, copper, and cobalt. All these are required to running the metabolism of the plant. And then at the end you also need the carbon source as sucrose. So you take these components, you mix them together and in an appropriate composition, in appropriate quantities, and then you autoclave them under an autoclave, and you also include the agar so that it gets solidified. So once you do that, you also going to get the solidified media. Now what you do is you take the surface sterilization of the X plant. So in this case, for example, if you see we have taken a leaf. So what you do is first you do the surface sterilization. First you take the leaf, then you first sterile the surface by using a detergent called 2-in-20. Once you do the washing with the 2-in-20, then you do the washing with the water, so that the excess 2-in-20 will be removed from your X plant. Once that is done, then you also should do the surface sterilization by the other reagents. So all these, what are the other reagents? Then you use the 70% alcohol. That 70% alcohol is antimicrobial. So it is going to kill all the bacteria which is present on the on the surface of the X plant. Why we need this surface sterilization is because the surface sterilization will ensure that once you put this X plant into the appropriate culture media, all these the microbes which are present on the surface of this particular X plant will not grow, because the growth of the microbes will be faster than the growth of your X plant. After every sterilization events, you definitely have to wash the X plant with a distilled sterile distilled water. This will ensure that the X plant is not going to be get affected by your sterilization procedure, and this is going to be for a very short period. For example, you are putting the leaf into the 70% alcohol for some time, but then you are washing it again back with the sterile distilled water, so that the alcohol which is present onto the leaf is going to be removed. Otherwise, this alcohol is going to damage the leaf and its integrity. Then ultimately you sterilize the X plant using the mercury chloride. Mercury chloride is a poison. So that actually will ensure that whatever the little traces of bacteria or fungus is present in your X plant is going to be removed. And after that, you are going to remove the mercury chloride again by using this sterile distilled water. After this, you are going to use a borer, and this borer actually you are going to use onto the leaf to cut into a small disc, and these discs will be removed from the borer using the forceps. All these instruments what you are using in the cell culture in the plant tissue culture are sterilized previously. Then you take this small disc and put it into the media or inoculate into the media, and that actually then you allow them to grow onto the media. And this media also contains different types of plant hormones. So that plant hormone, different types of plant hormone also allows the growth of this X plant onto the media, and eventually what you will see is you will see a undifferentiated mass of tissue onto the within the test tube, and that actually can have a potential to give rise to shoot and root. After that, you change the microenvironment of root into the callus, so that it actually will develop the shoot onto the top and as well as the root at the bottom. And then you can take this into the tiny plantlets which you have developed into a test tube, and you can put it into the pot, and you can cover this with the polythene, and so that it should still be not in good enough to get its own immune system working, so that it can actually fight with the foreign pathogens and other things. So you initially want to help the plant to get acclimatized. So, this polythene is porous. So, it is good enough to get the oxygen and carbon dioxide, but it is not allowing the a huge quantity of pathogen or the bacteria to come. And then once you acclimatize this plant, then you can place it into a pot, and then you can take this plant to outside, and that will let this plant to be acclimatized for the outer environment or to the natural environment, and that then the complete the plant tissue culture is done. So, what are the advantages of vegetative propagations, and why we are doing the artificial as well as the natural vegetative propagations? So, what are the different advantages and why we do actually the vegetative propagation is, so vegetative propagation is the most selective way of making the new plants in those cases where the plants do not have the ability to form the seeds. So, inability to form seeds. Classic, there are many examples where the plants are could not be able to make the seeds because they have several types of hurdles in getting the either the pollination working or the fertilization working, and those kind of plants are normally being propagated by the vegetative methods. Okay. Number 2, the plants are forming the seeds, but they are forming very small number of viable seeds. Okay. So, small number of viable seeds means the plants which are falling under this category is that these plants actually the fertilization is either incomplete or it actually not very efficient. So, in these cases what happen is there are many fruits which are being formed, but the fruits are devoid of either viable seeds or the seeds which are very good in terms of withstanding the extreme environmental conditions. So, what happen is as soon as you take the fruit, it goes into the micro environment or it goes out of the environment, and because of the very harsh conditions the viability of the seed get completely compromised. Then number 3, you are actually exactly the same, like poor viability of seeds. Okay. Number 4, vegetative propagation actually is making as we discussed in the previous time also, the vegetative propagation is like making replicative copies of the same plant. So, there is no mixing of traits, there is no mixing of traits at the genetic level, and because of that it actually keeps the purity of a plant species. So, it maintains the purity. So, if the plant is very good, it does not get mixed up with the different qualities, and as a result the good qualities are getting diluted. So, it actually maintains the purity of a particular line, purity of plant species. Number 5, with the help of plant tissue culture, you can actually generate thousands of plants or millions of plants in a very short period of time. Means it is not as per the life cycle or the developmental cycle of the plant. You can take the plant, you can inoculate into the thousands of test tubes, and you can get thousands of plants in a very defined time frame. So, you can get the disease free plants in adequate quantities, as many as you want. So, you can get unlimited number of plants. Even the purity of the plant species is maintained if you are really interested to bring the traits or particular traits into a plant species, that actually comes by the grafting. So, vegetative propagation actually allows you to maintain the purity of a particular species, but at the same time it also gives you the flexibility of bringing the new traits with the help of grafting. It also gives you that same potential even in the plant tissue culture, where you can actually change the conditions in such a way that actually plants start producing the different kinds of or different some type of the micro phytochemicals in a adequate quantities. So, with this we are going to conclude the asexual reproduction. Now, we will move on to the next topic, and that is the sexual reproduction. So, the sexual reproduction, as the name suggests, require the presence of the two different types of sexes. So, every organism who wants to continue with the sexual reproduction should have the two different individual, and they should have a male individual and a female individual, and both of these male and female individuals produces the gametes, and these gametes are actually then fused and give rise to the new organisms. And that is why for the sexual reproduction the presence of male and female individual is important. In many cases these male and female are present in two different individuals, and those kind of organisms are known as the unisexual organism. Unisexual organism, where you have separate male and female, whereas you can have the organisms where you can have the both male and female organisms, male and female individual on the same individual, and those are called bisexual organisms. So, here you have male and female on the same individual. Having the bisexual organisms, the advantage of bisexual organism is that they do not need the other individual in the close proximity, and that they can use themselves to reproduce whenever they want to reproduce by the sexual reproduction. But apart from this, the sexual reproduction requires both the male as well as the female to mature enough so that they can be able to produce the gametes, and this whole process is different in the different organism. For example, in the case of plant, they have a phase where they come out from the seed. So, as you know that the plant originates from the seeds, and then they go for the adult plant. The adult plant which actually can go the flowering, and this phase from seed to adult plant is known as the growth phase or the vegetative phase. Once the adult is formed, it actually start doing the flowering and this is called the reproductive phase. So, this is the phase when the flowers are formed in the these plants, and then they can start the sexual reproduction. Okay. This vegetative phase from seeds to adult plant can vary from months to years. So, this can vary from months to year. Similarly, the reproductive phase could be annually or could also span from several months to several years. In some places, so it can go from several months to years. Sometime the reproductive phase is also very much linked to the season. For example, some plants have the reproductive phase in the winter season, and so this is also seasonal. Sometimes some plants may have the reproductive phase in the winter, or some plants may have the reproductive phase in summer. Why some plants have a reproductive phase in summer and some plants have a reproductive phase in winter is mainly because the duration of the day and light, and there are other factors also which could be responsible for controlling their seasonal activities. Whereas, in some cases the reproductive phase actually occurs either in several years. For example, the classical example in this case is the bamboo tree. So, in the bamboo it actually flowers every 50 to 100 years, and it happens only once in a lifetime of the bamboo plant. So, so that is why the reproductive phase is almost 50 to 100 years long, and so that is for the plant. For the in the case of adults, or in the case of animals, from the birth to the adult stage can vary can be divided similarly into two phases, from the birth to adult stage and then from adult to the reproductive stage. Okay. So, I think we have discussed this. So, this is called growth phase, and this is the reproductive phase. Okay. For example, if we take an example of in the case of humans, it goes from 0 to 18 years. In the 18 years, the person becomes adult, and then it goes from 80 to 40 years as the reproductive phase in the human being. And there are animals which are actually doing this in terms of seasonal variation. So, some of the animals are their reproductive phase is only active during the summer, and in some cases it is only active in the case of winter. So, just like plant, there are seasonal variation, or there are seasonal activities of reproductive phase, and that is only because most of the invertebrate animals or the vertebrate animal actually require a optimal temperature as well as the environmental condition for the developmental phase, or for developing the egg into a particular organism or particular baby. So, that is why there is a seasonal variation, and that is how they prefer to have their babies in a particular season. Apart from these, so these are the two different classes what we have discussed, but apart from these, every sexual reproduction has the several things in common. For example, the sexual reproduction starts with the male and female, and both of these male and females are mostly being the diploid. So, mostly they are the diploid, okay, and both are the male and female are having the male sex organ and the female sex organ, and the male sex organ actually produces the male gametes and female is producing the female gametes. So, as I said, most of the organisms are diploid in their adult stage, but within the male sex organ they produces the male gametes by a process known as meiosis. So, because of that the male gamete or the female gametes are haploid, and these two fuse to form the egg, and that is the diploid stage, and this eggs develops into the adult. So, this is the actual schematic diagram or schematic schemes in through which the female sexual reproduction occurs in any organisms. So, if you see this scheme, what you see is it has three distinct phases. One is this phase where the male or the female actually utilizing their male or female sex organs to produce the male gametes, and that phase is called as pre-fertilization stage. Okay. And then you have the fusion of the male or the female gamete, and this stage is known as the fertilization stage. Okay. And once after the fertilization, what you are going to get? You are going to have the initial egg or the zygote, and from the zygote you are going to develop the adult individual, and that is the post fertilization Okay. So, if I summarize this, in the sexual reproduction you have three phases. One is pre-fertilization stage, second is fertilization, and third is the post fertilization. And every phase has a distinct function in terms of in terms of controlling the process. For example, the pre-fertilization phase is required to precisely do a meiosis step as well as the development of male or the female sex organs, so that they produce the adequate quantities of the gametes, and they also ensure that the gametes are produced in accordance to each other, so that there will be a high probability that these two gametes fuse to each other. For example, it's not happen that in the male plant there is a flowering, and the female plant it is actually there is no flowering. Okay. So, it actually ensures that the two different individuals actually synchronize the development of the male as well as the female gamete at the same time, so that when there is a male flower or there is a female flower, they both mature at the same time and produces their respective gametes, so that there will be the optimal fusion or optimal fertilization. Then the fertilization event is where the male and female gamete come closer to each other, and there are features which are present on the male gamete, there are features which are present on the female gametes, which make them for optimal which make them to do this process very optimally. So that, for example, in same of the cases, the male gametes are present within the, for example, in the case of plant, the male gametes are present within the pollen grain, and these pollen grains actually are not motile. So, they actually take the help from the environmental as well as the other factor present in the environment to carry from one place to another place and reaches finally to the female gametes, and that is how they do the optimal fusion and produces the zygote. Once the zygote is formed, the post fertilization events start. So, once you have a zygote, it develops into the adult, and that also has the several steps with the predefined developmental stages and as well as the predefined programming. So, that there are different steps which are involved from the zygote to formation of the adult or the all these three events which we are going to discuss now onwards in a subsequent lecture series. Thank you.